We're going to move now to our second set of three speakers here, starting with Katherine Roche, Dr. Katherine Roche, who's going to talk about the study of GRIN2 C-terminal domain missense variants and truncating mutations. Thank you.
Okay, I want to thank the organizers for including me. I always learn so much at these meetings and appreciate interacting with the other scientists, but also the families. My lab for many, many years has studied NMDA receptor expression and trafficking, and I'm going to talk, in this short amount of time about how that interfaces with GRIN disorders. So for decades, I guess, we've studied postsynaptic proteins, both the glutamate receptors, but also other components of the postsynaptic density. So for two neurons to communicate, it's important to have the glutamate release from the presynaptic terminal, but of course you have to have the receptors and the signaling complexes correctly targeted and aggregated at synapses. In this is a simplistic depiction of a synapse, but still I've drawn circles around some of the different classes of proteins that we've studied through the years. So not only NMDA receptors, but also adhesion molecules and signaling molecules and some of the cytoskeletal elements. And I should say that everything kind of got turned on its head in my group about 15 years ago when it became clear with the advances in sequencing of human populations that many of these proteins that we've studied for a very long time are implicated in neurodevelopmental disorders, and specifically that de novo mutations, the rare variants that are highly penetrant can actually be pathogenic. So we really shifted our research to that. Now, we've already heard about NMDA receptors, so I won't belabor this, but just to point out a few things on this depiction, NMDA receptors are heterotetramers, 2 gluon 1s, typically 2 gluon 2s. Our group has focused on 2B and also 2A. These gluon 2 subunits are highly expressed in hippocampus and cortex. We've spent many years studying the C-terminal domain, which the structure has been solved for NMDA receptors. They unceremoniously lop off the CTD. We don't know the structure of it. So we've been curious if we can get any insights from the rare variants into the function of the CTD. But I want to make clear that as more and more of the patient populations are sequenced with GRIN disorders, the CTDs are relatively tolerant. So we have studied a certain of the subdomains where we have clusters, small clusters of rare variants that are in probands. This includes the PDZ binding motif at the extreme C-terminal domain. We're just wrapping up a project, multi-year project on that. We also have a project on CAMK2 binding site. So there are certain subdomains within this big 600 amino acids that definitely can be pathogenic. However, in general, it's less so. Now, the GRIN disorders, we know that GRIN 1, 2A, and 2B are highly implicated, and at this type of meeting, you'll meet parents and children with variants in all of these genes. However, they present differently. And just to make the point, GRIN 2B, almost all of the affected individuals will have intellectual disability or developmental delay. Not all of them will have epilepsy. It's kind of the converse for 2A, where the vast majority will have epilepsy, and many, but not all, will have some sort of developmental delay or intellectual disability. And certainly some of the parents here will be quick to tell you that as they meet other children or families with children, some of them will have the exact same mutation and it presents differently. So it's certainly a challenge to try to address these different mutations. If you go to the Broad site, you'll find that for 2B, the vast majority, I think we've heard this already a couple times, and we'll hear it again over the next few days, are missense mutations, the vast majority. This is a challenge because they can be one-offs or just a couple of probands with the particular variant. and they can be gain of function or loss of function. So once again, it presents a challenge as to approaching therapeutics. However, and missense mutation, I won't go through this, but it's just a single nucleotide change that results in a single amino acid change, and the proteins are made up of amino acids. So that shows how sensitive an NMDA receptor is, that one amino acid change can, and if it's in a sensitive region, lead to dysfunction. There's also truncating mutations. Now, truncating mutations are premature stops, so it's typically, it can be a point mutation, it could be an indel, but essentially what it does is it causes a coding codon to be switched to a nonsense, and it stops. So you end up with a shorter protein, if you end up with protein at all. And to be honest, you can actually group these together, or I would make the argument that you could make a pretty good argument that they could be thought of as haploinsufficient. For the most part, you would have to do the experiments, but for the most part, these are going to have messages that are not stable, and you lose an allele, so you have half as much protein. Now, I'm not going to go through this in any great detail, but just to remind people or inform people that there's this quality control mechanism. If you have a premature stop, the mRNA, the cell detects it and tags it for degradation. This is why the vast majority of premature stops result in the loss of an allele, the expression of one allele, and you end up with half as much. However, you don't know, this is a complicated field unto itself, and sometimes it's cell-specific, and it's certainly transcript-specific. You have to do the experiments. So about a year ago, we put together this. As we've already heard, the databases are incredibly important. We rely upon them, but they're also kind of a mess. So ClinVar, I wouldn't swear each of these is a distinct patient or affected individual, but the point I want to make is just from a quick look at the databases, you'll see that GRIN1 and GRIN2D have many, many truncating variants identified in patients in Nomad and far, far fewer in ClinVar, consistent with it being very well tolerated. So GRIN1 and GRIN2B seem to be unaffected by dosage, or at least at the level of this type of analysis. 2A and 2B are the opposite. You find many, many affected individuals that have truncating mutations. And once again, this is just us taking a look at Searchlight, and I cannot implore the parents and the families to put the data into Searchlight. It really simplifies scientists studying your particular variant because they collate the data, they get the consent. Anyways, it's a great way to drive forward research on the variant of interest. This is just to point out, just looking at Searchlight, which is a subpopulation of variants, you see there's many, many truncating variants in 2B. So we've decided, especially because I enjoy trying to interact with the community, that it would be important to try to model some of these. The two that we're working on right now is arginine 847. This is very much a collaboration with Steve Trenalis and Hongji Yuan at Emory. They've made this mouse. I'll show some data from it. And we now have the mice at NIH, and we're breeding them. And so we have some data to show you on that. We have made, it took a long time, had some issues. We finally made the mouse with the 519. Now, why did we pick these two? We picked them because 847 is quite interesting, seems to be a hot spot. The best we can tell, there's probably 20 affected individuals that we could get in contact with in the United States and Europe. And some of them are at this meeting, some of the families. So it's more than the one-off. It seems to be a sensitive residue. And 519, I would say we're in the area of 3 or 4 affected individuals that we could get contact with. The other thing that's interesting about these is they're an arginine to a stop. I'll show a slide in a minute. This is not random. There's different potential point mutations that can lead to premature stops. This is the most common. The CGA to a TGA is the most common in many different disorders, not just GRIN disorders. The cartoon, the depiction here on the left, what we've done is we've indicated the truncations. 847, you get all of the protein, just missing the tail. 519 is far earlier in the protein, whereas you would only get this portion that is really a soluble portion before the first transmembrane domain. So these are experiments in my lab, just Hey Young's here at the meeting, Sehun's not, with the cDNA expressing these truncated proteins in heterologous cells. So this gets around mRNA decay. That's nonsense-mediated decay wouldn't function in this. It's just really to look at the protein stability. And we see that the full length migrates, and you see it on a Western blot at the correct apparent molecular weight. And then you see the truncated protein here for 519, the same for 847. So it expresses fine, these two truncated proteins, and there's nothing inherently destabilizing about those. But we made the mouse, I should say Steve's group made the mouse with 847. So we're now characterizing the het, which represents the patients. And in this case, on the left, they've been sending us brains, and they've been doing assays themselves. We do the kind of expression protein work. And looking at the message, we see that there's no change. So this is consistent with there not being nonsense-mediated decay. The 847, I should mention for more aficionados, 2B, the last exon is almost the entire CTD. It's an unusually large exon. And typically, although you have to do the experiments, if the truncations in the last exon, it is more likely to escape nonsense-mediated decay. This is actually 847 is really in the penultimate one. It's not in the last one, but it appears to escape it. So this is an in vivo model, endogenous promoter, het. We then we being Sehun, did in my lab, looked at the analysis and found there's about half as much protein. So you can see here the full length protein, there's about half as much. That's what you would expect because one allele has the truncated one. You see very little of the truncated species here, but it is there. And he's done more quantitative, he's looked at PSD fraction, it's the same, and he's looked at more quantitative dot plots, and it appears that the 50% that should be truncated, there's far less expressed. So we would estimate about 80% of the truncated protein is destabilized and degraded, or at least we don't detect it. Now, Steve and Hongji have done work at Emory on these mice, and they find that there's functional consequences. AMPA-NMDA ratio is altered. They've looked at spine density and CA1 pyramidal neurons, and they see a reduction in the spines, in particular mushroom, which is interesting. Those are typically mature synapses or represent. This is consistent with when we use C-terminal variants that do have a pathogenic phenotype, and there's less 2B on the surface or less 2B protein. A lot of times we see in culture fewer spines, fewer synapses. So this is consistent with that. He's also run some behavioral assays and finds that these het mice exhibit increased anxiety. This is a maze in which they don't want to go. Well, the animals can spend equal time either in the covered portion or the open portion, and the het mice prefer covered portion, which is interpreted as increased anxiety. They've also done seizure threshold assays, and they find that there is reduced seizure threshold in this particular assay in the het mice. It's all very much in progress. None of this is published. Now, once again, I'm going to circle back. Why are we interested in these truncations? Why are we interested in these particular ones that are CAG to a TAG? We're working quite closely with Chris Ahern, who's at University of Iowa. He's really been spearheading efforts into trying to develop tRNA therapies that can basically replace the nonsense mutation with the arginine and allow read-through. Obviously, this would be fantastic because there's so many truncating variants. It's already been licensed to Tavard Biosciences. They are focused mainly on Dravet syndrome. So as I mentioned briefly, this is an approach that could potentially rescue premature stops that are in other conditions as well. So what have we done with that? Just going back to the cDNA, so these are the truncated and hex cells. What we found is you see the wild type is the appropriate size, the truncated protein smaller, but if you co-transfect with this tRNA, you can rescue it. You can get the read-through and get the full length. And more recently, Hey Young, who's here, has done the same thing with the 519 and shown that this approach, at least in these heterologous cells, you can get a rescue. So where are we going now? We're, like experiments in progress, putting viral tRNA expression into the mouse model, see if we can rescue the protein, the functionality, and the behavior. We've also, in my lab, made the human iPSCs that are HETs that express these truncated for 519 and for A47. Oh, and this I put in as a reminder to myself and to you that we also have made a mouse model with 2A, and I believe it's 681. It's the same thing, the same mutation that theoretically could be have the same approach. We picked it based on multiple ClinVar hits. If there's anyone here, I should talk to see if anyone knows any of the families of those affected individuals. So the take-home message, GRIN disorders result from highly penetrant rare variants. They're mostly missense. People spend a lot of time and effort trying to figure out their loss of function, gain of function. However, I think you can group together many of these truncations and potentially affect a larger population of affected individuals. And I think I thanked people as we went along. Hey Young's here at the meeting, and Sehun's not, but they're the ones that have driven the research. That's it.
Okay, wonderful. Now I'd like to welcome Dr. Amy Ramsey, who's going to talk about glucose regulation in GRIN1 mouse models. Thanks.
Good morning, everyone. Thank you so much for inviting me to talk. This is the first time I've talked about this topic, and the slides are still a bit rough, but let's just dive in. Okay, I wanted to point off a few things starting off. This has always interested me. NMDA receptors are the only receptor that I know that can bind to multiple amino acids and requires them to be activated. So glutamate and glycine are not just neurotransmitters, they're also building blocks. And beyond that, at higher concentrations, you can get activation with aspartic acid, with homocysteine, and with even L-serine. So, these genes have existed for a long, long time. They're found in plants and bacteria and in animals that lack a nervous system. So, NMDA receptors might have something to do with energy sensing and metabolism. So, keeping that in mind, when we move to animals that have organs that manage energy, we see NMDA receptors in peripheral organs, including the pancreas, which is really important for glucose regulation. And they're also, of course, found throughout the brain, including the hypothalamus, which is kind of the brain's region to sense energy demands. And so just as a little reminder, there's kind of two main hormones that the pancreas will secrete. So if the brain senses low blood sugar, it will cause the pancreas to release glucagon, which then liberates it from the liver so that you can get blood sugar to be released. And then if blood sugar is high, oh, do I have this wrong? Yeah, okay. When it's high, like when you eat, then that causes the pancreas to release insulin, and then insulin will take that sugar and either put it into tissues or will store the extra in the liver as glycogen. Okay, so keep in mind just the pancreas secretes glucagon and insulin for right now. Okay. So we've known for a long time that these mice that have low levels of NMDA receptors, because they have low levels of the GRIN1 mRNA, that they weigh less. So this is their weight over age, and here we've got it divided by males and females, and then this is their weight here at this last time point. So you can see that the males weigh less than their wild-type litter mates. And the females do too. Sorry, this is the low end, but I think we've increased it since then. And anyway, it's held true for many years. So more recently, we did an analysis of, kind of what these animals are made of: their fat, their lean muscle, and their bones. And what you see is that the mutant animals have less fat tissue and more lean tissue, and that's seen in the males and also in the females. So the other thing that we did to kind of start to look at what's going on with their metabolism is to put them in these metabolic chambers. And these chambers, the animals house there for no more than 24 hours. And after we let them settle in, this chamber is sealed up so that you can measure oxygen consumption, CO2 production. So that can give you an idea of their respiratory exchange rate. Also, it measures their activity and the heat that they're producing. So what we have over here is their activity with the mutants in pink, and here we've combined males and females. But this is mostly males in this assay, I think. So you see that they're more active during their wake cycle, which is at night, and also during their sleep cycle. They are producing more heat, but it's not as remarkable as their increase in activity. But this part right here is really interesting because this ratio can indicate what kind of respiration is going on. So are they oxidizing carbohydrates or fats? Because carbohydrates have this respiration quotient, or CO2 over O2, of 1, whereas fats, when they're oxidized, have a quotient of 0.7. So a lower RQ would mean more fatty acid oxidation as an energy source. So these lean animals that don't have a lot of fat are using fat as an energy source, as you can see, because they have a lower RQ. I find that really fascinating. So I wonder, this made us remember a paper that we'd we'd published a while ago, where we were testing omega-3 supplementation, but also omega-3 deprivation in their diets. And we didn't see a lot of benefits from omega-3 supplementation, but we did see some problems in diets that they had lots of fats, but they just didn't have omega-3 fats. And we saw these are Kaplan-Meier survival curves. So you're seeing males especially start to die, pretty young age. So actually I think that we put them on this diet at weaning. So, they get the diet at 3 weeks and then they start to die. So by the time we were testing them for behaviors, most of the males had died. And we also saw this for females, but clearly there was a sex difference there. So this is going to become important. Sorry, this is not working. I don't know if it's a battery or, the pointer is working, but okay, thank you. Okay, so that male-female thing becomes really interesting. I think we might have skipped can you help me? I'm sorry. With this, it's back one slide. Okay. So this male-female difference became really interesting to us. And so when we're looking at glucose, again, trying to understand what's going on with their metabolism, one of the things that probably you're familiar with if you've been tested for diabetes is that you're given glucose and then they measure how quickly your body clears it. So that's an indication, your body should take that glucose and put it into your tissues and put it into your liver. And it does that with insulin. So, so what you'll see over here is a spike in insulin, or a spike in glucose when you give glucose in the blood. And then the body will clear it. So you actually see that it's getting cleared pretty quickly in the KD mice. So this isn't a problem of, the glucose not getting into tissues. It does seem to be cleared. And we have started to do some of these same tests with the patient variant mice. So I don't have a lot of data about them. But it looks like with the males, we do see, again, Q536R is kind of a mild loss of function. Y647S is a more severe variant with mixed loss and gain phenotypes. But we can see that it is being cleared pretty quickly. However, when we look at the response to insulin, so here instead of giving glucose and measuring glucose, you're giving insulin and you're measuring glucose. So here we give the insulin. You can see that the resting glucose is lower, particularly in the KDs, and also to some extent in the Y647S. And then when you give insulin, it drops further, and then it recovers. And in the case of these Y647S, it dropped to incredibly low levels, which can cause death, and in this case did cause seizures in the animals. So it looks like these animals are very sensitive to insulin. It doesn't take a lot of insulin to get a drop in glucose. So to me, that says that the insulin receptors are not desensitized, and that's what you would see with type 2 diabetes, where you always have high levels of glucose. The insulin receptors kind of become numb to the glucose, and so in this case, they're super sensitive, which would suggest that maybe there's not enough insulin floating around normally, so whenever they see it, they can really do something with it. The other way that you can measure glucose regulation is by causing exercise, which will normally liberate glucose from the liver, and you should get an elevation in glucose, and that's what you see normally in the wild types, but we did not see this same elevation in the KD mice. So that aspect of glucose regulation is also impaired. So looking at the pancreas where insulin is produced, you can see, just looking at these pancreatic islet cells, which is where the insulin-producing cells are located, that they're smaller in the male mice. And this really was not seen so much in the female mutant mice. We don't have data yet on the patient variant mice, but this was really striking when we performed immunohistochemistry staining for the insulin secreting beta cells and the glucagon secreting alpha cells, which these are beautiful structures in the pancreas. You can see that they're really small in the KD males. And we also, these pictures don't really show it, but there were a lot where there was a lot of disorganization, and you'd see alpha cells really in the middle of, and kind of throughout, so there were not these beautiful islands that they should be. So then these were stained, we used a semi-quantitative method to look at intensity. So within these areas, how much insulin was being produced, and there is less here in the males and females. And there was also less glucagon intensity in the males, but not really significant in the females. Okay, oops. So what could be causing this? Sorry, I'm nervous to do this. Okay. Interestingly, I found this paper looking for things that can cause pancreatic islet cell, developmental problems. And what they found in this paper here is that if you kind of remove the norepinephrine that is landing on these islet cells during development, then you get these smaller islets and also some disorganization of the islets as well. And this can cause decreases in the amount of insulin and in insulin release in response to glucose. So it could be that these effects are due to changes in innervation. And of course, we think about NMDA receptors in the sympathetic nervous system being important. Okay, I also wanted to point out this interesting paper from 2014 where they did proteomics of the serum and saw that a related hormone, insulin-like growth factor, was down, leptin was down, and glucagon was up. So there are some changes that have been seen in some of these in some of these regulators of energy, and so just kind of bringing us back to this picture, it does seem that there is a change in really in insulin and in the cells that produce insulin, and that doesn't necessarily lead to what we would call diabetes, but I think it does suggest some changes in glucose metabolism. So just to summarize, the KD mice have reduced weight with less fat, more lean body mass. The metabolic chambers indicated greater use of fatty acids as food energy source rather than carbohydrates. The islets are smaller, fewer in number, with some disorganization. I didn't show you this, but you'll have to take my word. Lower insulin and glucose levels in the serum, faster clearance of glucose, and higher sensitivity to insulin, and exercise does not increase blood glucose as it should. Future questions: is this seen in patients? I really want to know. Is this developmental? So even if we fix it in an adult animal, fix NMDA receptors in an adult animal, would we fix these smaller islets that we're seeing? Can this be corrected with medicines, with diet, with gene therapies? And also, could glucose or insulin be useful biomarkers for clinical trials? They're awfully easy to study. Okay, I want to end by thanking the lab, especially Tatiana Lapina, who really spearheaded this, has run this from beginning to end, and I'm so incredibly grateful to her. We have a great crop of undergraduate students, and Emily is just joining our lab, a former undergrad, soon-to-be graduate student who's here. So is Lyra, and so is Sylvia. So look for them. And I also want to thank my collaborators. Lionel and Maya worked with Tatiana to do this work. Okay.
Wonderful. Thank you so much. We're now going to have our last speaker of our morning session. So please welcome Erica Nebitt for talking about NMDA receptors in neurodevelopment and seizure pathogenesis.
So, hello. Thank you for having me here today. Can you hear me? Hello. I'm very excited to be here. I'm an MD PhD student at Stony Brook University, where I'm currently working on my PhD in Lonnie Wilmoth's lab, and I'm going to talk a little bit about part of my thesis work studying the role of NMDA receptors in neurodevelopment and seizure pathogenesis. So my interest primarily lies in epilepsy and seizure disorders, as epilepsy is increasingly becoming the most commonly diagnosed chronic childhood condition. So in the neurotypical nervous system, as we all know, activity exists in a standard balance between excitation and inhibition. Seizures then arise when there's an imbalance in neural activity, and this can be due to an increase in excitation, a decrease in inhibition, altered membrane excitability of neurons, or a combination thereof. Oops. Okay. And so, just pausing there for a second, childhood seizure disorders are usually due to mutations or variants in proteins that participate in membrane excitability or synaptic transmission, including NMDA receptors, which is the focus of my work and the lab's work. Now it won't advance. Okay. So, to date, as we've heard about before, a multitude of disease-associated variants have been identified in the GRIN genes encoding their corresponding subunits of the receptor. And like others here, I collated data from publicly available sources and grouped variants as pathogenic and putatively pathogenic based on known patient data and pathogenicity. And so what I'm showing here is for pathogenic and putatively pathogenic variants, I've focused in on missense variants, and I assess the proportion of positions along the gene for which missense variants have been identified. And so this shows that for genes like GRIN2A and GRIN2B, they're much more likely to have these missense variants as opposed to GRIN1 and GRIN2D. And going off of that, the percent of patients with seizures who have variants in these genes differs per gene. So GRIN2A and GRIN2D tend to be highly associated with seizures in patients. So I'm interested in studying how these genes contribute to seizures via their loss of function. So I'm solely going to look at their loss of function and look at seizure development. And to do this, I'm using zebrafish as an animal model. And so zebrafish are a very powerful tool here because they develop rapidly and they have a high degree of NMDA receptor conservation. So all the fish I'm using are aged 6 days post-fertilization. Here you can see an image of one of my fish. This is a pretty central time point in neurodevelopment, and it's also an age used in reference brain registries, which I'm interested in taking advantage of, so I'll have consistent time points across all of my experiments. It's also important to note that zebrafish underwent an ancient whole genome duplication, so they have 2 paralogs, as you can see here, for almost every NMDA receptor gene. So when I'm talking about the loss of function of these genes involved in early neurodevelopment that I'm interested in, the fish that I'm using are loss of function for both functional paralogs. They don't have it at all. And then the wild types I use in my experiment are those most closely related to each individual mutant group of fish. Okay. So to study seizures, I first wanted to develop a pretty quantitative method to look at convulsive behavioral seizure activity in the fish. So I developed these convulsive seizure experiments in our lab where I take the fish at 6 days and I embed them in agarose, so hopefully you can see that they're head fixed in agarose, but then the tail is free floating in solution. So I can really look at that tail activity as a measure of convulsions. I'll record a 10-minute baseline, and then I'll put on a chemoconvulsant to measure seizure susceptibility. The chemoconvulsant I used is PTZ, or pentylenetetrazole, which is an antagonist of GABA A receptors and is commonly used as a model of acute seizures. And then I'll record a 30-minute experimental condition. I will run the videos that I take through Deep Lab Cut, as you can see here, and I'll track these discrete points along the fish, and then I can calculate the angle of tail deflection as my metric of convulsive seizures between this point on the body of the fish and this point on the tail, and I get an output trace that looks something like this. And then here, you can see a video of one of my fish where I've put on PTZ, and you can appreciate that it'll start to have these really large level convulsive seizure-like events, and this is what I consistently see in my experiments. And then when I run the video through Deep Lab Cut, it's able to fairly effectively track the various points I'm interested in, including, as you'll see, these points on the end of the tail, even when the fish is moving a lot and the convulsions become pretty high in amplitude. Okay. So then from these experiments, I get activity that looks like this. I have baseline activity that is very low level, very low in amplitude, and it's very repetitive and stereotyped. And we think that's just swim-like behavior that the animal is experiencing. And it's very different from what I see during the experimental condition when I put on PTZ. I start seeing these large level, high amplitude tail deflection events interspersed with minimal to no activity. So we think these are our convulsive seizure events. And then in this red box, I'm highlighting what we have come to term as an individual seizure event. And then kind of expanding on that, this is all data from wild-type animals so far, and you can just see a zoomed-in version of an individual seizure event. There's a lot of seizure-like convulsive activity going on in there. And then I went on to analyze these, and to do so, I really wanted to detect convulsive seizure activity and not baseline level activity. So I began by running a series of experiments in fish treated with 0 millimolar PTZ or vehicle. I analyzed the average maximum deflection of their tail, and I used that value averaged across fish as my threshold. So that's what I'm showing here. I set these thresholds for my analysis, and I detect the activity outside of that threshold. And you can see when I then treat the animals with 10 millimolar PTZ, which is my fairly standard concentration, I see an increase in all of these parameters that I'm able to extract from the data. So we see an increase in the event frequency of the seizures. We see an increase in the duration of the individual seizure events. We see a significant increase in the average maximum deflection of the tail, which is really that metric of convulsive seizure-like activity. I also see a significant increase in the probability that the animal is in a seizure event state throughout the experiment, and I see an increase in the activity frequency during individual seizure events. So during one of these events, the animals, when treated with PTZ, have a lot of activity throughout. And then I started doing these experiments in the mutant fish. And so far, I've only done them for GRIN1, GRIN2A, and GRIN2D loss of function fish. But my data shows that the GRIN1 and GRIN2D lacking animals experienced increased seizure frequency, which you can see here and here. But the GRIN2A fish surprisingly did not show an increase in seizure frequency. But this shows that the loss of function of GRIN1 or GRIN2D at the level of these convulsive seizures coincides with increased seizure susceptibility. What starts to become interesting is when I look at all the different parameters that I'm looking at, the phenotypes aren't always the same for the same animals. So here, when I look at the activity frequency during seizure events, I only see a significant increase in the GRIN1 loss of function animals and not the GRIN2A and GRIN2D lacking animals. So the prevalence of these seizure phenotypes seems to vary across my examined parameters. And so here, I just wanted to show this summary slide of the 3 groups of mutant fish that I've looked at and some of the parameters that I have examined, and you can see that the loss of function of these subunits differentially alters the parameters I'm looking at. And it almost develops a phenotypic fingerprint of the seizure activity in each of these groups of animals due to the loss of function of these genes. And this is really interesting because our ultimate goal is to uncover the etiology underlying the loss of function of these genes, mechanistically, neurodevelopmentally. And we're interested in taking that information, growing this data set of this kind of phenotypic fingerprint and integrating a machine learning approach to ultimately, hopefully, be able to screen variants identified in human patients, identify kind of that phenotypic fingerprint, and hopefully be able to infer or intuit the underlying pathology, seizure pathology associated with those variants, and hopefully bring treatments to patients that much faster. So that's the ultimate goal here, and it's very encouraging thus far. Some of the other experiments I've been working on are recording electrographic seizures or local field potentials in the animals, and so now I'm really interested in focusing in on seizure-like brain activity, whereas convulsive seizures can kind of integrate both brain and spinal cord activity. And so the most important task for me to do here is to stabilize the animal, so I'm only getting brain activity in my recordings. And as these experiments have never been done in our lab before, I first turned to the literature to develop a protocol, and I did a lot of troubleshooting that I'm going to walk you to. So previous experiments using this technique to record field potentials would just embed the fish in high concentrations of agarose to keep them stable. I did this at a high agarose percentage of 3.5%, and the animals were very stable, but I could still see even like skin twitches in the skin of the animal around the electrode, and I was definitely picking that up in my recordings. So I moved to paralyzing the animals, and I first started with alpha-bungarotoxin, which blocks nicotinic acetylcholine receptors, and for some odd reason, I had a lot of trouble getting consistent paralysis of the animals. So I tried another paralytic. I moved to pancuronium, which is also often used in studies like this, and it's an antagonist of nicotinic acetylcholine receptors, And I was able to successfully paralyze the animals, but then I wouldn't see seizure-like activity until over an hour after treating them with PTZ, which was odd and also diverged from the literature. So I tried one more paralytic, which was tubocurarine, also an antagonist of nicotinic acetylcholine receptors. And this one so far has been working really well. The animals are paralyzed. I'm able to get consistent recordings about 10 minutes after washing on PTZ, and tubocurarine is also more specific for nicotinic receptors, whereas pancuronium has some muscarinic binding properties as well. So, turning back to my experiments, I take my animals, I paralyze them with tubocurarine at 6 days, I embed them completely in agarose, you can't really see it here, but this fish is embedded, and then I insert an electrode into the optic tectum, which is the region from where I'm recording, and I record baseline activity, I put on PTZ, and then I record experimental activity in the presence of PTZ, and I get events that look like this. And so here is a more detailed look at what my activity looks like. When I record it, the baseline activity is very flat, low level, some brain activity, but not that much going on. And then in the presence of PTZ, I detect these large level events that are, I believe, to be seizure events induced by PTZ, and this is in wild type animals. So far, I've only done these electrographic experiments for the GRIN2A loss of function animals, but my preliminary data does seem to indicate that these fish have increased seizure susceptibility. And this is reflected in the parameters of event duration, the probability of being in an event state, and activity frequency during individual seizure events. So this shows us that these electrographic recordings are a very valuable tool because they can detect seizure activity that may not necessarily be prevalent at the level of these convulsive seizures. So that's exciting and encouraging, and I'm looking forward to doing it in the other animals. And then to just kind of summarize a lot of my work and what I've talked about, I've seen so far that fish lacking NMDA receptor subunits, or the three that I've looked at, GRIN1, GRIN2A, and GRIN2D, seem to show increased seizure susceptibility, likely increasing an imbalance in neural activity in the brain. And then to quickly touch on some future directions, I'm interested in carrying out immunostaining for phospho-ERK to detect these brain regions that may be contributing to seizure development. So ERK is extracellular signal-regulated kinase, and what happens in neurons is that when there's a calcium influx into the cell, it activates this downstream signaling cascade, leading ultimately to the phosphorylation of ERK. And so phospho-ERK can and has been used as a marker of active neurons, and this is what I'm interested in doing. And here I just have an image of one of my fish that I've stained with an antibody against phospho-ERK, and I've imaged it, and it's labeling these active neurons. And I'm ultimately really interested in doing this for all of the mutants, mapping them onto a reference brain to carry out a consistent fluorescence intensity analysis across these discrete brain regions. And I'm interested in seeing if there are brain regions in the mutant animals that inherently have increased activity levels, kind of suggesting regions that may be contributing to increased seizure susceptibility in them. Oh, well, that brings me to my acknowledgements. So I have lots of people to thank, especially my lab, the Wilmeth-Shea lab, the Sirotkin lab, who Howard Sirotkin is here with us, for helping us collaborating on all things zebrafish, my thesis committee, the program in neuroscience and the medical scientist training programs at Stony Brook, and then some of our funding sources that I also have here. And thank you all for listening.
Perfect. Thanks so much. I'll invite our speakers back up for a quick Q&A.
Very nice talk. This is on. Very nice. Yes. Okay. Very nice talks, everyone. I had a question for Amy about the pancreas. And so do you think that the defects you see are autonomous to the pancreas, or is this really a peripheral nervous system issue in terms of what NMDA receptors are doing?
That's a great question. And I think this one, I think it's nervous system. And we are testing that with an Advil Cree, which, so the knockdown is an animal where you can cut out the mutation and kind of restore it back to wild type. So it's like an inducible rescue, conditional rescue. So we can use an Advil Cree, which is expressed in the peripheral nervous system. And when we look at those animals, we see some improvements. But that work is really preliminary. Tatiana's been doing that, and she's actually mainly been doing it to look at like sensory issues. But you know, that same peripheral nervous system that's, you know, controlling sensation is also, you know, the motor component. So there's a motor component that's Advil Cree is anyway, that's what I think. But it's a good question, and we don't really, I don't really have that answer yet.
Okay, follow up. Do you have any guesses or data on any N2 subunits that might be involved?
I don't remember. Like the people in my lab would know this, but I think that you know, the single cell atlas probably has that information. Definitely, if you look, you'll see kind of by intensity of expression that after the brain, the pancreas kind of has the most. So, but I always look at GRIN1, and I don't always look at the other grins, you know, but I'm embarrassed, so probably we can get that answer.
You and then you.
I have a question that might be a stretch, and it might just be my lack of understanding, but, you know, keto diet is used a lot in a lot of channelopathies to help control seizures. And this is for Dr. Ramsey. And the mechanism isn't very clear as to how that happens. So I was really interested when I heard your talk. And I just can't help wondering if there's some kind of link that you see that's maybe between the glucose dysregulation and like the efficacy you see in keto diet and other pediatric epilepsies.
Absolutely. That's what I think, and that's kind of what led us to really look at the glucose. There's a whole story that we have around looking at ketogenic diet, and one of our students, Megan, has been giving beta-hydroxybutyrate to one of the patient variant models and saw a reduction in seizures. So I think that giving an alternate energy source in the form of fat is likely to help. At least in the mice, it does. And it doesn't have to be a ketogenic diet. I think that just, you know, just a higher fat diet would probably help.
Hi, morning, Amy. Great story. I was wondering, I'm thinking, how long do you follow the mice into adulthood? Because I'm thinking of two patients I have, one with a GRIN1 and one with a GRIN2B, who developed diabetes type 2 quite early, too. Okay. And they have proven they are on metformin. And I'm thinking, what I'm seeing in the patients when they're young, we very much recognize this lean body. These are children, have a great appetite, but are very lean. Yes. And a subgroup, I feel, has this switch to obesity type 2. And I have one GRIN1 and one GRIN2B that I know by the top of my head. So I was wondering, it would be interesting to follow the mice to see, because also when you think about diabetes from the diabetes field, like I think when the islands become smaller, it feels to me like they're losing.
Yeah.
So
I thought it was more type 1, and I don't understand why they're clearing glucose. so well. Like they clear it really well. So it's not a pure kind of, we have aged them. We never see them get fat. I am wondering, are they males or females, your patients?
Females,
Okay,
both.
okay.
And the thing is just that on a mechanistic base, I'm just thinking about like also the hypothalamic. Like I
Yep.
think it's, yeah.
Of course,
I haven't
it's super
figured it out
important.
yet, but it's.
It is very important. And again, you know, this is where the genetic models can really help dissect that, you know, to rescue it just in certain hypothalamic nuclei and separate out what we're seeing. Is this due to changes in appetite, changes in sensing, you know, versus kind of execution, you know? So yeah, there's
And
still a lot.
the other thing I thought, like, I don't know if it's in the database that we have with Professor Lemke, that we can think whether maybe hypoglycemia in childhood is pretty common. So maybe you would expect that in GRIN1 we would have more often episodes that the children deteriorate when they have an infection or something. But I don't know. I know of some parents that are saying when the children are ill,
Yes.
afterwards, they are a brief period, they're better. And then they start going back to the the disruption of the behavior again. Yes, that's what we thought of ketotic, yeah.
Okay.
So in more GRIN patients, you hear this story.
Yeah.
The children will come in with just a regular infection and whatever, and then afterwards they sometimes have a developmental like a step up, and then the behavior is way better than normally, and then they go back to baseline and behavior again.
I really hope that this encourages, first of all, it's great to see you. I'm so glad to see all the clinicians and everyone. I really hope this gives you the ammunition to do some CGM, you know, to do some continuous monitoring. I would really love to see that data, and I understand that it's invasive, but it would, if we could connect that to seizures, I think it could be really powerful.
Yeah.
It's so much easier to measure in humans than in mice, you know?
No, I mean, with the sensory glucose, I don't think it's a big issue. It's we will figure it
Yeah,
out.
I'll do it. I've done it myself. It's not that bad.
Hi, very nice talks. I have a question for Katherine. So potentially we'll go more to the anatomy. You show a very nice phenotype with the decrease in the mushroom spines, right? So the question is whether also is presynaptic terminals also there is a decrease or a reorganization, and where do you think that it starts?
Right, so we just don't have the answers to that yet, but I agree with you that that's quite fascinating, that there's such a significant decrease in total spines, and it seems to be entirely in the pool of mature spines, which, as you know, is consistent with mature, developed synapses. But the smaller villipodia-like are unaffected. So those are Steve's data, and this is all very much in progress, so we really don't have any data beyond that, but I think it's an interesting thing. We see similar things in culture with a lot of, any sort of mutation we have in the CTD that's pathogenic, and 2B that affects surface or total expression affects spines in a negative way. So it seems to be that NMDA receptors, if you decrease them, you're going to have less of that, and that affects overall synapse number. That's all we know at this point.
Thank you. And also for Erica, hi. You show a very nice video with the very rocking tail moving. So do you think that this is a special distribution of the receptors in the fish that may affect that more? And what are your thoughts in terms of, or what are the expectations between the brain versus the tail?
That's a really good question, and I honestly haven't thought about it very much, but I think that's a good thing to keep in mind. I know that there's definitely a different spatial distribution within the brains of the fish across all the GRIN subunits. I'm not that familiar with what's going on in the spinal cord, but I'll look into it.
Thank you.
Yeah, I have a question too.
Because I thought it was unusual that the 2A, if I caught what you presented, did not show as many seizures, but then it had an increased susceptibility for seizures. But this is quite different than what we would have expected from the human presentation. Do you have any ideas as to the differences between the human condition and the model?
So that's a great question. I was very surprised when the data didn't show a difference at the behavioral level. The data from the recordings in the brain is very preliminary, but it is showing a difference, so that's encouraging. My biggest guess is as to what is happening is that perchance the deletion of 2A is not as big an insult developmentally. It's generally expressed a little bit later in development, so maybe it's not having as detrimental an effect. So you're not seeing it at the behavioral level, but then you are seeing it when you kind of get closer into the brain. Or there's something going on that we're picking up from the spinal cord that's kind of confounding and mixing up the data.
I just had a question about immobilizing the fish. Are they short on oxygen? Like, how do you, like, they're okay?
They're okay.
Yeah, okay.
To my knowledge, they're okay. They don't tell me. But the agarose is at a concentration that should be porous enough that they're always covered in fluid on top. They're never completely dry out, so it should always be diffusing through the agarose into the fish. And then at the end of every experiment, I make sure everyone has robust blood flow. Otherwise, I don't include that data. Okay. But I think they're okay.
Thank you to the three of you and for all of our speakers today. Thank you very much.